was going to try to cover all of Peyronie's disease, and then I saw it was a 20-minute time slot. And the other factor was two things. It seemed foolish to try and cover Peyronie's disease when we have two leaders in the field who developed the guidelines, Dr. Kara and Dr. Miner here. So why would I tell them things they already know? Instead, I am going to challenge the dogma in the spirit of this conference named Innovations. And if you want to take me out back and beat me for heresy afterwards, that's okay. That's acceptable. All right, so these are my disclosures. So, all right. One of the things that always bugged me about Peyronie's disease is that we don't know why it starts. We don't know how it really happens, but we know what it is. We see what it is. So similarly, I'm going to draw the analogy. These three children have facial features that distinguish them, and they all have one common thing. Shout it out. What is it? Down syndrome. So Down syndrome children classically have almond-shaped eyes, upward shifted eye shape. They have one singular hand crease. They have frontal bossing, kind of flat faces. And when you see that face, you're going to have several associated medical conditions. So from 50 paces, you see that face and that child might have a heart defect, might have cataracts or hearing loss, is definitely going to have short stature, probably a thickened neck, hypotonic muscles. So what I'm leading to here is there's a pattern. Long before they knew it was trisomy 21, which was discovered by a French scientist, Dr. Lejeune, in 1959, which is really, if you think about it, like a generation or two ago in medical terms, they knew that this facial feature went with this medical syndrome, right? So similarly, we know the pattern of Peyronie's disease. We know it's typically Caucasian men in their 50s and 60s. They usually, maybe 50% of the time, will develop erectile dysfunction, and that erectile dysfunction might be more difficult to treat than others. There's an active phase where they're developing whatever deformity or plaque that they're going to develop, and then there's a plateau phase. The active phase may be associated with pain. The classic teaching is like micro trauma, but what does that mean? 2/3 of patients in my clinic have no recollection of event. They just woke up one day and this started happening to them. Maybe a third remember like some kind of incident, and then everything started to happen. We know that they form a plaque. Not all patients, but a lot of patients form a plaque, and that's a spectrum between cartilage and bone. And we know that it's associated with some other features: diabetes, radical prostatectomy, and potential Dupuytren's contracture. Now, why am I so rabidly passionate about this pattern and what we do and don't know? So in my former life, before becoming a medical professional, I actually spent 1.5 years in a tissue engineering lab that took mesenchymal stem cells and pushed them to cartilage and bone, which set me up for this. And then I also spent 1 year in an immunology lab. And if you think about Dupuytren's, Peyronie's disease, an exaggerating scarring response and deposition of collagen where it shouldn't be, kind of sounds like an autoimmune disease, right? So a couple of the things that really bother me about this are we're really missing the forest for the trees here. So when thinking about Dupuytren's, we think, is Peyronie's disease, is it autoimmune? Because in a study of 415 men with Peyronie's disease, 22% of them had Dupuytren's. And then in another study where 85 men had Dupuytren's, 26% of those patients had Peyronie's disease. So we know there's a higher coincidence of those two conditions than should be in the general public. The other thing that's interesting is, we tell these patients, because of the guidelines, and please don't shoot me for impugning the guidelines a little bit, we tell them, okay, we see that you have Peyronie's disease, just wait till it forms, take some ibuprofen, and then come back when it's really done its damage, which is really frustrating for patients. Firstly, that they waited so long to see a doctor and we tell them ibuprofen. But secondly, tomorrow we're going to hear some really great cancer talks. What if I had kidney cancer and I went to my doctor and the doc said, yeah, I see it on the scan, but here's the thing, we're going to let it do all of its damage. We're going to let it metastasize. We're going to let it spread and do like maximal damage. And then when it's done carving up your body, that's when we're going to intervene and we're going to give you systemic chemo to mop up the mess. That's essentially what we tell Peyronie's patients. We say, like, wait, you're still like forming this plaque. Let's wait till we get to the end of this when you've stabilized, and then we'll fix the deformity. So not only is there a time delay, but we're not intervening when we have an opportunity to do so. Another thing I'm going to challenge. So the classic teaching is dorsal plaques, right? Plaques that happen under the neurovascular bundle along tunica albuginea and just above those cavernosal bodies. But here's the thing that never made sense to me. If you really see Peyronie's disease, then you know that's not true. In this paper published in 2018 in the Journal of Sexual Medicine, we look, I call it kind of like the 32 flavors of Peyronie's disease. Plaques are lateral, they're ventral, they're dorsal, there's bidirectional curvature. Some people have volume deformity loss. Some people, like, an indentation, a hinge. Other people don't have that. Some people don't even have a palpable plaque. So I don't know that these plaques are all happening where we're saying they're happening. Also, is it just the tunica albuginea? Raise your hand if you've ever put in an IPP in a patient with Peyronie's disease. If you've ever had to pass those dilators, you know it's not that simple. And if it was just the tunica albuginea, it would be that simple now, wouldn't it? It wouldn't affect the cavernosal tissues. Do we have that right? I'm not so sure. We used to think that the plaques were exclusively cartilage, and now we know in recent years, there's this nice study, my friend at VCU, 71 patients, they looked at it histologically and showed that almost 40% of plaques are ossified. So, okay, sometimes we need to get outside our silo. Did you know that there's a disease process in the orthopedic world where if you bump into things, you form heterotopic ossification? So like your muscle forms bone in it. So we're taking the penis and the tunica albuginea, a place that shouldn't form bone, and they have some kind of trauma, and then some spectrum between cartilage and bone happens. Okay, so here's what we do know. We know that there's some kind of inciting event, which we don't know what it is. We know that there's some kind of inflammation and that pathway is probably mediated by TGF-1 beta. Going back to medical school, it's the inflammatory pathway that leads to fibrosis. We know that matrix metalloproteases are upregulated. We know that that leads to collagen deposition in a disordered way, potentially also bone. Basically, it's a bunch of extracellular matrix that shouldn't be there. Again, I think of it kind of like a keloid, right? Like we've all been cut in our lives, but not everyone forms a keloid in response to injury. So is it a two hypothesis like cancer? Are these people genetically predisposed and then something happens to them and they develop this deformity? Forgive me for this. I don't have a PhD, but I'm going to give you a little crash course in the TGF pathway. Okay, so there's two receptors. Try not to go to sleep. I know it's postprandial. Two receptors. TGF-beta 2 binds with TGF-beta. That phosphorylates TGF-beta 1 receptor. Then they go to SMAD 2 and 3. Those get phosphorylated and activated. They bind to SMAD 4. Then they translocate to the nucleus, and what they do is they act as a transcription factor. So they fundamentally alter the way the genes are expressed inside the cell in this TGF pathway. Now, this was just, the TGF superfamily pathway in nature, but this applies to Peyronie's disease. So people are doing some interesting work, and I'm just presenting this as what could be done, a couple of representative studies. So in a lab in Germany, again, published, this one, the first study was published in EU and then JSM, they looked at plaques, right? They took Peyronie's plaques, and then they took tunica albuginea from control patients who were having plications for like congenital curvature, so patients who ostensibly didn't have a trauma response. Then they took some tunica albuginea, they isolated the fibroblasts from these, they incubated it with TGF-beta for an hour, and they watched what happened. And what happens is if you look at the Western blot and the time increments, 15 minutes, 30 minutes, 60 minutes, that the SMAD2 and SMAD3 are quantitatively increased. They're expressed more, and they translocate to the nucleus more rapidly. Okay, so what's SMAD? SMAD is a key signal transducing molecule that basically is like the worker bee that carries out the information of TGF-beta. So it's the downline messenger that initiates the fibrosis pathway. Okay, so then in a follow-up study, this one, they looked at Peyronie's plaques again versus the control patients, 7 and 3 in this study, they did hematoxylin and eosin staining. So in this first look, if you look at B is actually normal. B is a patient who does not have Peyronie's disease. And what you see is spindle-shaped fibroblasts. You see normal collagen deposition. In A, you can see clearly disordered collagen deposition around nuclei. And so, like, there are some just basic stuff that you can see. And then in C, you see formation of bone, lamellar bone formation. So none of this should be in the tunica albuginea in the penis. So again, they take fibroblasts out of control patients, Peyronie's plaques, and now they put them with TGF-β, but for a longer time. They call it like long-term, bathing them in recombinant TGF-β. And they look at those same SMAD proteins, okay? And they look at SMAD7, which is an inhibitor of the pathway. And then they look at MCP, and I'm going to have to cheat here, monocyte chemotactic protein 1. Monocyte chemotactic protein 1 is a chemokine that recruits monocytes from the bloodstream and brings them into the tissues, and then it activates that fibrosis pathway. They show that these are activated with TGF-β, but there's no differential between these two, between patients who have Peyronie's and patients who don't have Peyronie's. Now, here's the one thing that I have problems with in both of these studies. Both of these studies took plaques from patients who were in the plateau phase, 12 months after their plaque had formed. So I'm going to argue really the action is in the active phase, right? That's when really all the differential gene expression's probably going on. Segue a little bit. We have the genome now. They've done genome-wide association sequencing, whereas there's about 10 different genes associated with Dupuytren's contractures. There's one, WNT2, that shows a locus and association with Peyronie's disease. You can also look at differential gene expression. So these are individual patients. One patient with Peyronie's disease, one patient who did not have Peyronie's disease, taking the tunica albuginea, distilling out tissue culture, getting those fibroblasts, and then looking if they're exposed to TGF-β, like what happens, what gets activated, what gets downregulated. And then they have this nice Venn diagram where 11 genes are upregulated. So in that study, they're identifying potential molecular targets to suppress the fibrosis reaction downstream, right? They're showing us like this is what's happening in real time. Okay, so if I were you, I'd be sitting in the audience and I'd be thinking, cool, that's all really interesting. Why don't you go figure it out? Okay, firstly, give me money if you want me to do that. It's not cancer. I have applied to the local grants 5 years running and I get shut out because cancer trumps a curved penis every day of the week and twice on Sunday. Secondly, recruiting and identifying patients in the active phase. It's actually really tough because they haven't developed the full deformity. They're in that process. Now, I will say, and I'm not allowed to say brand names, but the bent carrot commercial has been very helpful in getting active phase patients into my office. Secondly, okay, patients come into your office and you need tissue, right? These were plaques, right? So I'm saying, hey, can I just biopsy your penis? Like just a small little bit. It's for science. It's not going to help you, but it'll help other patients. That's cool, right? So as you can imagine, all of you who have penises, no one really volunteers for that. Then additionally, not only do we want to biopsy their penis, but we want to do it in the active phase. And as I referenced, the smart people in the audience who wrote the guidelines tell me not to do that. So patients are getting taken to the OR in the plateau phase. And again, referencing those prior studies, 12 months in, I would argue that really all of the mRNA and molecular changes and interesting things that are happening in the active phase when that patient is remodeling their tunica albuginea, and that's what we need to look at. Lastly, who serves as the control group? Who's going to donate a little of their tunica albuginea when they're perfectly healthy? Again, just give me a little piece of your penis. It's okay. We just need to look at other people who have a disease, right? So these are some challenges in Peyronie's disease research. I have had trouble getting some money. I have had trouble getting people to give me pieces of their penis, et cetera, et cetera. So these are some beautiful pictures of my OR, and this is how I got into reconstruction and sexual medicine. These operations are challenging. They're technically amazing, elegant surgeries. But those are people's genitalia that I dismantled. The most important part of their body, some would argue, although clearly the heart and the brain are quite important. But the penis is very important to men. Patients in my office tell me that over and over again, 9 years on repeat. I don't know if you know, but penises are very important to men. And I say, yes, I've heard that once before. It's a defining sense of self and identity, and this disease deforms them, disfigures them, emasculates them. You use whatever terrible diction you want. It is deeply disturbing to them. And instead of trying to prevent it and put ourselves out of business, we have these great, cool surgeries we do after the disease process has done all its damage. So what do we need to do instead? Instead, we need to join forces and have a multi-institutional collaborative effort at tissue banking. And then we need to do molecular assays on these. We need to look at not just the plaque, but the underlying cavernosal tissue below the tunica albuginea. Because if it's inflammatory cytokines and, transcription factors, that cavernosal tissue is bathed in the same milieu that the tunica albuginea is. We need to be doing genetic microarrays, looking at the upregulation of mRNAs that we could have as targets to suppress. We need to look at epigenetics because there are familial portions, like there have been studies showing familial passage of Peyronie's disease. Now, not all of them. Some of them are probably sporadic. But like VHL and kidney cancer, we've noticed that some families have a familial predisposition to develop kidney cancer. And so Peyronie's disease has the same thing. And we need to explore associated disease processes. So, you'll hear on Sunday about chronic pain and how that's associated with other disease processes from me. If patients have a predisposition to form scar tissue in their penis, where else are they forming scar tissue? Are there other things going on that are linked? And we talked about the Dupuytren's contracture, right? That's just one example. What if it's linked? There's some data to suggest HLA-B27 with rheumatoid arthritis and ankylosing spondylitis might be linked. So those are things we need to be looking at rather than just perfecting our very cool surgeries. And with that, I'll take any questions.